Here is what the plate reader gave to us. You can see the grid that resembles the shape of our 96 well plate. The software automatically colors in different shades of blue based on the absorbance. So it looks pretty, but it's not easy to analyze in this form. What I've done for you is created a second sheet called Organized Data. You'll see in your Excel file there are 4 sheets total. This one has a couple more because I want to show you what things will look like as you finish. Here's the Organized Data tab. What I've done is created a well ID based on the column and row for every well and the absorbance. So now we have all 96 wells listed in one column. Your first step is to use the plate map that you filled out in lab to record what information or what kind of sample was in each well. So your plate map should look something like this. We see in well 1A, that was our 0 standard, standard 1 with a concentration of 0. Well 1B was our second standard with a concentration of 0.1. So based on your plate map, you're going to start filling out column B. See if I can make both of them show up at once. All right, so I'm going to use a pretty specific way of IDing all of these wells. If you follow this exact convention, it'll make it a little bit easier down the road when you have to sort your data. Remember, we had 2 sets of every control, every sample, and every standard. I'm going to refer to the 2 different ones as A and B. So I'm going to label this as the first is what the A stands for, the first well of standard 1, which had a concentration of 0. Right, looking at our plate map, we can see that the second well, IB, with standard 2 with a concentration of 0.1. Right? I would keep filling these in. If I look at my plate map again, I can see that my first control, the low control, shows up in 1H. I'm going to name that A. Again, it's the first of the controls, QCL, and then our high control, QCH. After that, in 2B is our first sample, sample number 1. And again, I'm using the specific lettering system to help me sort later. I'll say A-01 and then B-01. Both of these were sample 1. Next well, A02, B02, A03, B03, and all the way down. If we look at our plate map, we can see our last 2 duplicates for 39 were in 11F and G. That's going to be down here, right? So A-39, B-39. Then I start with my second set of standards in 11H. I'm going to call that B standard 1, again with a concentration of 0. Followed by B standard 2, that concentration with 0.1. All right, so once you actually complete all of this, you should have all of column B completed with this labeling system. It will look something like this. The reason that we used A's and B's is it's going to make it much easier to collapse our duplicates into a single value. We want to take the mean of every pair. So standard 1, we want to average the value of the A at the beginning of our plate and the B at the end of our plate. Right, so the easiest way to do this, there are several different ways in Excel, or if you're using another software program, that's fine. Do what works for you. The reason I set it up with this lettering system is we can pretty easily sort the data by this ID or concentration column. I tell it that my list has headers. The header I want is ID or concentration. And now you can see it's sorting all of the A values together and all of the B values together. Right? Why is that helpful? I'm going to copy all of the B values I'm going to bring them up here instead and paste them in cell 2D. Right. I want to recopy the header row over. Now what I can see is I have both values for the high control QCH, both values for QCL, both values for each standard lined up the same. And then when we get to our samples. Okay, so we've got these B's here because we moved them already. We can go ahead and get rid of them. Right, so now we have that data organized and sorted where we have one row with both of the duplicate values. The next step is to calculate the mean, the average of the two values that make up the duplicates. To make things a little bit easier, I'm going to hide these two rows. Right, and if you look over at the averaged data column, or I'm sorry, the average data sheet, one thing to note, I have an extra column in here that you can get rid of, the one labeled absorbance. We don't actually need that. Instead, we just want mean absorbance. In addition to that, as you can see on the instructions, we want to calculate for each pair of the two duplicates standard deviation and that coefficient of variation. Ultimately, we'll calculate mean percent bound, but that comes a little bit later. Right, so again, mean absorbance, standard deviation, and CV for coefficient of variation. If we copy those over, it's going to give us some guidelines of what we need to calculate. This is why it worked well to do this particular naming system. We're going to be able to take advantage of the formulas built into Excel. I'm going to ask Excel to take the average of these two cells. Since I hid those two columns, technically they're factored in as well, but they're not numbers, so Excel ignores them. Right, here's the average of the two cells, the two wells that I had for the high control. Right, we also want to take the standard deviation of those two values. Here's the formula in Excel. Again, I can highlight both of them. All right, and lastly, the coefficient of variation is calculated by dividing the standard deviation by the mean. We usually express that as a percentage, so in Excel, the easiest thing to do is select that percent icon. You can also multiply by 100%, but just do one or the other. And maybe we would like one more decimal place there. Right, the beauty of setting it up this way is we can now copy and paste. right? And I would actually want to drag this all the way down, but just so you can see here, we now have an average value for each of the two duplicates that we have for our two controls, our standards, and starting to see for our samples. You would want to pull it all the way down. In the end, it would look something like this, right? I filled in the percent bound column. We're going to talk about how to do that in a second.